to develop more flexible statistical models that can analyze a wider range of traits in, extreme environments, for example. And if our genomic predictions in particular are successful, then we can help reduce the breeding cycle. Now, one thing that my lab has become really big on, especially in our research, is simulating traits. So given a set of genome-wide marker data, can we randomly select them, SNPs to be quantitative trait nucleotides and form the basis of the trait. And my former postdoc, now assistant professor Samuel Fernandez, helped me develop this R package called simple phenotypes. So our motivation for developing this is that multi-trait quantitative genetics models have become really widespread. And these can be potentially important because they can help us identify, pleiotropic genes or genes in linkage, that might control multiple traits. And for certain traits, it can help improve prediction accuracy. So we realize that simulating correlated traits will really help us rigorously test how useful they are.
Because if we're simulating traits, we know the truth. We know what the actual genetic architecture of the trait is, right? And so therefore, we can directly assess true and false positive identification rates. And so, thanks to the heavy lifting of Professor Fernandez, we developed this R package called Simple Phenotypes. And it's available on GitHub, and Professor Fernandez still updates the GitHub maybe on a monthly basis. So here's one example where we use simple phenotypes for part of the research. So this is the work of my former PhD student, now Dr. Matthew Murphy, and basically, he was interested in applying variance GWAS approaches to test for G by E. So here's a picture of the United States, right? So let's say that we wanted to grow some maize in the state of South Carolina over here. Now, South Carolina is not where the Corn Belt is in the United States, so perhaps it's not the best place in the country to grow corn, right? So therefore, breeders might want to select against G by E, so you can breed for resilience in growing environments. So you want to breed for stability, and then, I don't know, a really awesome place to grow maize in the United States, Champaign-Urbana, Illinois, where I'm from. You might want to select for G by E so that you can select for responsiveness in really good growing environments. So maybe resistance to new biotic stresses or new management practices. So regardless if your specific breeding objective is selecting for G by E or selecting against G by E, it is important. And maybe we want to find G by E loci. So the approach that Matt was looking for was finding an alternative way to indirectly identify G by E loci and not have to deal with the ensuing multiple testing correction. So what he came up with was, why not use variance GWAS approaches? And this is just a quick illustration of what a variance GWAS would do. So on this left figure, let's say we have the null hypothesis that a particular locus does not have an association with your phenotype. So we got our genotypes on the x-axis, phenotypes on the y-axis. The null hypothesis is true, so we see the same population mean of our phenotypes, regardless of little a, big A, little a, big A genotype. So usually, if we're fitting a standard model like in the linear mix model, for example, we might test for an additive association between a capital A allele and the phenotype. And such statistical approaches assume constant variance. So we might see a very similar distribution of phenotypes about the means, right? So a variance approach is you're testing for differences in your population variance across the three different genotypes. So such a locus is called a variance QTL, and if you run a variance GWAS using such an approach, it's a variance GWAS. So Here's a really simple illustration of why we thought that maybe a variance GWAS approach could be used to look for G by E. So let's consider these two environments, environment 1, environment 2. We have this locus over here with this effect size. We have this same locus with a different effect sizes. Knowing something about the environment tells you what the effect size is. Therefore, this is a G by E locus. And look, if we just simulate traits of these two different environments, plot genotype on the x-axis and phenotype on the y-axis, lo and behold, the total variance of the trait increases for each additional capital A allele. And this illustration illustrates why a G by E locus might manifest itself as a variance locus. So we went ahead and did a simulation study and a real data analysis, and then ultimately Dr. Murphy got his PhD. So the reason why we wanted to do this VGWAS approach was the multiple testing correction is less severe. We're only testing, we're only conducting a single hypothesis test at each locus. And so we did a simulation study where we simulated G by E loci from real marker data so we could quantify true and false positive rates. And then we did a real data analysis where we looked at flowering time in maize across 2 mega environments. And then the 2 VGWAS models, 2 approaches we tested, the Brown-Forsyth test and the double generalized linear model. And then we also compared this to a unified mixed linear model just to have a baseline to compare to. So our simulation pipeline was intense, and I'm only going to show a subset of it. But if you're interested in it, check out this heredity paper from 2022. So we had genotypic data in Arabidopsis as well as in maize. And so we would simulate, one scenario is we simulated G by E, another one is we would simulate epistasis, and then another one is we would simulate differences in your population variances according to your genotype at loci. So proper variance controlling loci. And then we simulated multiple replicates of simulated traits and compared true positive detection rates across the 3 statistical approaches I discussed in the previous slide. So again, this is a massive study. I'm going to focus on the scenario where we simulated G by E, and I'm only going to focus on maize. So here's a picture of the results. Okay, so we simulated 2 traits, and we took the difference between our observed values at each trait, and that was our G by E trait. So here we simulated traits in both environments. Sorry, I didn't say that correctly. We simulated the same QTL in both environments, but their effect sizes were different. Here we only had a particular QTN present in one of the environments. And here we had another QTN present in the other environment. The y-axis on both of these is the true positive detection rate. And then the top graph is for the smaller sample size we considered. And then the bottom one, the bottom graph is for the maximum sample size of 2,815 individuals. And so in the bottom graph, we see that the bars are much taller, right? So also we see the orange and the blue bars, which are the 2 variance GWAS approaches, they can be quite high. So this is showing that when we simulate G by E, if the sample size is large enough, we can identify variance QTL. So we then wanted to see if we could obtain a similar result in real data. So we went to the Goodman-Buckler Maize Diversity Panel, consists of 281 lines and about 327,000 SNPs. And the really awesome thing about this data set, it has been heavily researched since 2005. So there are publicly available phenotypic data and genotype data. And so we decided to analyze 2 different flowering time traits, growing degree days to anthesis and growing degree days to silking. And then we had 2 quote-unquote mega environments. So we had 2 locations in the Midwest in the United States, indicated by these blue stars. and then we had these two locations in the southern United States indicated by these red stars. So this is how we obtained our G by E traits, okay? So we obtained Best linear unbiased predictions across each of the two, across the two blue environments and across the two red environments. And then we took the difference between BLUPs, and that was our Y variable. Now, I want to point out in this slide that we controlled for population structure and relatedness. So here's on our x-axis is our first principal component of a principal component analysis of our markers. And then our y-axis is principal component 2. And then the red dots are the 281 lines. And clearly these are clustering into groups based off of population structure. So indeed, yes, population structure is present in this data set. In this picture, we have a heat map of a Van Raden 2008 additive genetic relatedness matrix. And the colors correspond to the degree of additive genetic relatedness between individuals, right? So we're seeing that the colors are not uniform, the same color. So this is showing that we also have differential familial relatedness in this data. So we were duly diligent. We realized that subpopulation structure and familial relatedness could result in spurious associations. So both of these G by E flowering time traits, which I told you how we took them, how we obtained them, both of these were fitted to the unified mixed linear model that accounts for these two factors of spurious associations. And then the ensuing VGWAS approaches used the residuals, okay? So and then we used these residuals for the Brown-Forsyth test double generalized linear model. And bottom line, we knew there was population structure and relatedness in these models, and we tried our best to duly account for them. However, it wasn't good enough, okay? So if you read our crop science paper, you will see that we did identify statistically significant associations. However, we have 4 different QQ plots here. The rows correspond to the 2 different VGWAS approaches, and then the 2 different columns correspond to the 2 different traits we analyzed. Y-axis is our observed negative log base 10 p-values from our GWAS. X-axis is what we would expect these negative log base 10 p-values to be under a null hypothesis of no association. So we see that our p-values are severely inflated from what they would expect it to be under the null hypothesis. And then these lambda values, okay, these are genomic control, like the Devlin and Roeder 1999 statistic. And if our models are doing a good job of accounting for familial relatedness and population structure, we would anticipate these lambda values to be around 1. And look, they are severely higher than 1. So, and also recall, we were duly diligent. We accounted for population structure and relatedness. So this is essentially showing kind of a negative result. So this doesn't work yet in real traits. I guess that's what I'm trying to say. So our simulation studies show that both of these VGWAS models can identify QTMs with G by E effects. However, when we tried it on real traits, it didn't work because we still had inflated type 1 error rates. So it was kind of disappointing that this did not work, but we found out something, all right? And we figured out that if someone else in the future wants to pursue this, they can know where to look next. So accounting for population structure and familial relatedness in the traditional manner was not sufficient. So I think what needs to be done is this. From a quantitative genetics perspective, I think that I think that research needs to look at how population structure and relatedness affects not only the population mean parameter of our trait, but also how it affects the variance of the trait. And I think VGWAS approaches that are more sophisticated, albeit more computationally intensive, such as the hierarchical generalized linear model and the double hierarchical generalized linear model, which basically enables you to have a random effect in the variance part of the model and put a Van Raden kinship matrix in there, for example. Perhaps that can be a way to account for this problem that we discovered. Now I'm going to talk about a very brand new project. These slides is the first time I ever presented them, and this is so new that this paper was accepted into genetics on Saturday, like 3 days ago. So this is seriously new stuff. I also got to say it is interdisciplinary, okay? So all of these cool figures you see here was created by Dr. Edoardo Bertolini, who was supervised by my colleague, Dr. Andrea Eveland, at the Donald Danforth Plant Science Center in St. Louis. So we wanted to use genomic prediction to infer genetic architecture across species. And by cleverly tweaking your genomic prediction model and comparing your prediction accuracy to an empirical null distribution, for example, you can do stuff like that. So what Dr. Bertolini did was this. He took maize, so we got a maize plant over here on the left, okay? And he took two different subsets of transcription factors in the leaves and in the tassel. The first gene set was, gene co-expression. So this is a larger set of transcription factors. And then the second set was network motifs. And this is a smaller subset of genes. So what we did was we fitted three different types of RR-BLUP GS models. Each of the three of them had different subsets of markers as X variables. The first one was the whole genome SNPs, where we included all SNPs throughout the genome as an X variable. The second one was using markers in the vicinity of the larger set, the GC markers. And then the third model was these was markers in the vicinity of smaller subsets. Okay, so we did this in three different species. Whoa, sorry about that. This is so exciting that the mic gave me feedback. So we assessed the predictive ability where we would train our GS model in one diversity panel and evaluate prediction accuracy in the other panel. Then we did this again where Dr. Bertolini, figured out what the sorghum orthologs of all of these genes we identified in maize were, and then did five-fold cross-validation for markers within the subsets of these markers as well as genome-wide. And sorghum is more closely related to maize than our third species, rice, where we essentially did the same five-fold cross-validation study. And then what we did was we basically compared the prediction accuracies we observed of each marker set to a corresponding empirical null distribution of prediction accuracies of just randomly selected markers. Okay, so here are the results. And indeed, this is a really busy slide. So I'll tell you really quickly, the take-home message is the markers in the vicinity of the smaller set of transcription factors do a really good job of predicting our two traits in maize. But anyway, we got two traits here. Okay. And our left picture is for the smaller set of genes, and the right picture is for the larger set of genes. So we got two maize traits, tassel branch number and leaf angle. These purple arrows is the prediction accuracy of markers in the vicinity of each set of genes, okay? And then here are the empirical null distributions of randomly selected markers. And then here's the 95th percentile of these empirical null distributions. This is the prediction accuracy of the whole genome marker sets. So if we go back to the prediction accuracies of our NM marker sets and our GC marker sets respectively, we see that the prediction accuracies of the NM sets for both of the traits on the left graph, right, they are far beyond what we're observing for our empirical null distribution. So what we inferred from this is that these NM marker sets are doing a better job of predicting the respective traits than an empirical distribution of a random set of markers. So that was really cool. Now, here's the awesomest part of this work. When Edo got NM orthologs in sorghum and did a 5-fold cross-validation study and repeated this, if we look at the left graph over here, we see that the NM set is beyond the 95th percentile of the empirical null distribution. And if you think about this, if we were to construct a p-value, of this, the p-value would be less than alpha 0.05, right? So we have evidence that the NM markers, are predicting, leaf angle in sorghum better than chance. Now, unfortunately, we did not get the same result in rice. So, I'm kind of not surprised by that result because rice is more distantly related to maize than a sorghum is to maize. So, what we concluded from this work is that network motif genes in maize could contribute to the genetic architecture of leaf angle in maize and sorghum. So now my next thing I'm going to talk about is the omnigenic model. And don't worry, I'm going to introduce you all to it. And this is the thing I've really been excited about for the past couple of years. And now there's a project involving a lot of different groups and different sources of funding. But anyway, so here's the argument that I'm going to make. The seminal paper from Fisher in 1918 forms the basis of quantitative genetics, and that's a great thing, okay? So basically it says, as the number of genes controlling a trait increase, your effect sizes get smaller and infinitesimally small at the limit, okay? And also it says the genetic value underlying the trait approaches normal distribution. So if we were to make a graph, right, of the genetic architecture of a complex trait, your genes would be on the x-axis, and then the effect size is on the y-axis, and you see that the effect sizes get smaller and smaller and smaller. And the longer version of this presentation, I would show you that GWAS and genomic selection results, for the most part, affirm that this model is correct. However, a whole bunch of advancements have been made since 1918, right? So on the left side of this timeline is Fisher 1918, and then the right side is today, October 1st, 2024, right? So here are all the statistical advancements, right? I mean, pertaining to quantitative genetics, and this is an extremely short list. Like, Fisher 1918 was before ANOVA, before the mixed model, and before QTL analysis, before genomic prediction, Bernardo 1994, and and GWAS. And then here are all of the advancements that have been made in our knowledge of molecular biology. Like, we didn't even know how DNA looked in 1918, right? So, why don't we incorporate all we learn and, update models of genetic architecture? So one of them, one of the models that I'm really interested in testing is the omnigenic model. And here are two main observations that led to the development of the omnigenic model. And I'm going to show how I've seen them in my research, okay? So the first thing is that peak associations inadequately explain the total heritable genetic part of the trait. So this is an analysis I did way back in 2013. This is maize. The trait we were looking at was alpha tocopherol. Our x-axis is physical base pair position along the maize genome. And then our y-axis is a negative log base 10 p-value of a GWAS using the unified mixed linear model. So we see this peak at the end of chromosome 5. And I put forth that, that the gene underlying that trait did not completely explain the entire heritability of this trait. And then another observation, and this is a little more nuanced, and but associations that are identified in GWAS are not near a priori candidate genes. So on this picture, this is another paper that I did with my collaborator, Andrea Eveland. So the x-axis depicts a new statistic of importance that my former PhD student Brian Rice and I developed. And so we calculated it for genes that are in, for markers that are in the vicinity of MNA sites, not a priori candidate genes. And then we compared it to an empirical distribution of randomly selected sites, and lo and behold, we got a substantially higher importance metrics on the x-axis than the empirical null distribution. So, those two observations led to the development of the omnigenic model. So basically what it says this: if you consider a particular tissue, okay, all of the genes contribute to the genetic architecture of your trait, but all of these genes are subdivided into two bins. You have your core genes that directly regulate the trait, and then all the rest are peripheral genes that affect core gene expression. And so that was Boyle et al. 2017, and then Liu et al., both from Pritchard's lab, refined the omnigenic model to say that these peripheral effects indirectly affect the trait through transregulation. But anyway, so what this says, all genes that are not core genes are peripheral genes. And I stole my own thunder. Peripheral genes indirectly control the trait through transregulation. And then you have this concept of network pleiotropy, that within the tissue, peripheral genes indirectly control all traits. So that's great. But there is a variant to the omnigenic model that I'm really, really interested, because I think it could have an influence on how training sets are obtained in breeding. But this is from Ian Matheson 2021. So in the left bit over here, we have our phenotype, and then we have core genes that directly control the trait, and then we have peripheral genes and maybe environmental factors that indirectly control the trait. And then under the B part, we see that we have two different populations or subpopulations, and the peripheral genes within each of those two populations, they form different perfect storms of networks of trans effects that are indirectly controlling the trait. So what that means is, if we were to, say, fit a unified mixed linear model to a marker in the vicinity of one of these peripheral genes, we might see, a positive effect size indicated by that gray arrow pointing up in population 1, and a negative effect size of your marker if you were to fit the same model in population 2. So this led me to a set of competing hypotheses on the omnigenic model, okay? The leading hypothesis is that the omnigenic model is an accurate representation of genetic architecture across subpopulations. And if this were true, we would basically see K different subpopulations, and each of the different peripheral genes, with the blue genes, okay, would form the different perfect storm of a network. And this is indicated by these different colors. The competing hypothesis is that the omnigenic model is not an accurate representation, and we would see something boring across our subpopulations, maybe similar networks of peripheral genes. So if hypothesis A is correct, I would expect to see that the core genes have the same effect sizes across subpopulations, and peripheral gene effects will differ across subpopulations, and cross-population genomic prediction accuracy models will be low. And if hypothesis B is correct, I would expect to see core genes have different effect sizes across subpopulations. Peripheral gene effects will differ within subpopulations, and cross-population genomic prediction accuracies will be high. So I started investigating the omnigenic model in rigor. I did a sabbatical at the Roslin Institute in Edinburgh, Scotland, from January to May, and I studied in the Highlander lab, led by Dr. Gregor Gorjank. And they run the AlphaSim pipeline, which does forward in time simulations. So I combined AlphaSim-R with simple phenotypes developed in our lab to test this. So I started off with this founder population over here. I used burn-in, wheat parameters from the training paper indicated in that reference from 2023. So I ended up with 1,120 individuals. Now, to simulate population differentiation from this founder population. I'd be remiss. And then I use simple phenotypes to simulate an initial genetic architecture. So then I did two types of selection, two types of directional selection with different selection intensities, and then two corresponding sets of disruptive selection, and then two corresponding sets of stabilizing selection. And then I did forward in time simulations for 10 generations, ultimately resulting in 6 different subpopulations. And throughout this, I should use the present tense. I'm keeping track of how genetic architecture evolves. And then at the end of the day, I want to study the cross-population genetic architecture. So we have, for each of these 6 subpopulations, 1,000 SNPs, 250 QTL are our observed traits, and 3 replicates of founder populations. So here's the statistical model that we're using to simulate traits. And I'll tell you, in simple phenotypes, it's possible to simulate this. It might be even running right now. So our Y variable is our observed phenotype of our ith individual, okay? These terms indicated in red, these are additive and additive by additive epistatic effects of our 7 core QTL. I just randomly picked 7. 7 is my favorite number, so maybe it's not random. And then the terms in blue are additive and additive by additive epistatic effects of peripheral QTLs. And then the thing in purple is the interaction between our core and peripheral QTLs. And I'd be remiss if I didn't talk about the grand mean and the random error term. So the thing that is controlling the effect sizes, if you look at all of these terms, they are normally distributed with different population variances. So we varied the sizes of the variance of the, of the red, blue, and purple terms through a factorial study. A 3 by 4 by 4 factorial design. Factor A, we looked at the core additive versus peripheral additive effects. Factor B, we looked at core epistasis versus core additive effects. Factor C, we looked at peripheral epistasis versus peripheral additive. And then factor D, we looked at between core peripheral epistasis versus peripheral additive. And we simulated 3 replicates at each of the 192 treatment levels. Now, all of this work would have been basically a pipe dream if it weren't for the assistance of 2 of my colleagues at the computing center at Illinois, Dr. Weihao Ge and Dr. Joao Viana, who is here in the audience right now. So they actually parallelize it and coded it up and make it actually runnable. So prior to parallelization, it took 36 hours for running a single iteration of simulations, over 20,000 hours for analysis. So, right, we want to get this done in the next decade, right? And then Dr. Viana and Dr. Ge, they parallelized our master scripts. Now it takes 24 hours for simulations across all iterations, 6 hours to do analysis, and it's ran on this really awesome cluster, which has 70 nodes, 512 gigabases of RAM, a fast connection cluster, and it allows a fast interface between the user and the cluster. So this analysis that I showed you is currently running on the Illinois servers. Results are imminent. Oh, and there's not a big queue. And so I'm going to show some results from the summer in a simpler simulation study, because if if they pan out, they're really cool. So the major difference between the simulations that I showed or the pipeline I just showed now versus what I did in the past summer, I had only 3 subpopulations instead of 6. Okay, and this is how we evaluated our results. This is called the 4-trait trick. So here's where we simulated our trait. I already explained all the terms here. So that was our first trait. Our second trait is a core component where we only simulated the core additive and epistatic effects. The third trait was we simulated only peripheral additive and epistatic effects. And then and then our fourth trait is only core by peripheral effects. And basically, before I show you the gory details, we wanted to trace how these variance components changed generation to generation across subpopulations. And just to tell you what we did, we looked at the population variance covariances of our observed trait values, our observed genetic values, so all of these models minus the epsilon I term, and then also we looked at the observed breeding values. And, on the surface, that sounds like just the additive effects, but really it's additive and additive by additive epistatic effects. And Dr. Chris Gaynor helped us write this R script following this paper from William Hill in 2015 to get those observed breeding values. Okay, so these results, this is a really cool result, but it's on a really inelegant manner of presenting it. So if you have any suggestions on how to condense these next 6 slides onto one slide, I'm all ears. So here's one of the 3 types of selection, okay? Our x-axis is generations, 0 is the founder population, 10 is the 10th generation of selection, and then our y-axis is the log of the variance component of our genetic effects. So consider this to be a background, a baseline slide. And here we're looking at specifically the core variance components. Now, the cool thing about this is if the onogenic model is true, I would expect the core variance components to be relatively stable across our different subpopulations. So here's subpopulation 1, here's subpopulation 2, here's subpopulation 3. And the core components, the variance components, are not changing that much. Now, if the onogenic model works, I would expect the peripheral variance components to vary all over the place. So check this out. Now we're looking at the peripheral variance components and see how much they differ. And I know this is confounded by the fact that the ratios of the graph differ, but trust me, if you go to the PDF and slide through it really quickly, they do bounce all over the place. So this is a really promising result, one of the coolest things I've ever seen, to be honest, and we're going to undertake this next simulation study to see if it pans out. Okay. So, very last thing I want to talk about is a really excellent PhD student I have. I think you all know her, Talisa Floriani. And what she is looking at is the implementation of a random forest approach into GWAS. In this very week, Talisa is going to present these same slides at a meeting in St. Louis. So, we get... so this presentation is going to span 2 continents. So what she's interested in is rare variants, okay? So these are SNPs with minor allele frequency. We are saying they're below 0.05. In humans, rare variants are predisposed to individuals with diseases. So we need to have a lot of genetic diversity in order to capture these rare variants. And so therefore, Talisa is looking at the sorghum association panel, which consists of 4 different races of sorghum. And then this data set has 331 genotypes and about 116,000 SNPs. So her hypotheses for how machine learning can help with GWAS is that her favorite hypothesis is that the machine learning method will be able to associate rare variants with phenotypes. Her competing hypothesis is that it will not. So if this hypothesis is true, then we would expect to find at least one associated rare SNP. And then if the competing hypothesis is true, then the machine learning will not find any associated rare SNPs. Now, here are two excellent slides that Talisa created to help us visualize random forest, and I hope I do her slides justice. So at the top, we have our data set, right? And so then we're going to run multiple different decision trees, okay? Each tree has nodes, and that basically I understand that as a decision that splits our individuals into two different groups. And then at the end, we have a decision. So she is interested in looking at a mixed random forest approach, which is, basically it uses a mixed model that I'll show you in the previous page to make the decision process about how to split individuals into two different partitions. And then at the end of the day, across all the multiple trees, she will find the SNP that is the most responsible for doing the best possible job of splitting your data set into different groups. So this is the model that we're using. So our Y variables are phenotype of interest, and then we have our genotypes indicated by this matrix X, and then we have some function that models the fixed effects of the loci, and then we have the good old, individuals as a random effect, and then the Van Raden 2008 kinship matrix is used to estimate additive genetic relatedness, and we have our error term. I gotta say, Talisa, she basically learned a brand new software. She learned Python from scratch, and she coded this up all by herself, and I'm about ready to show some of her results. So she simulated traits under simple phenotypes. She did 3 different settings. I'm just going to show one of them to get the point across. So she randomly selected 4 different SNPs to be quantitative trait nucleotides, assigned each of them an additive effect of 0.5. So the settings under simple phenotypes she used was she simulated 1 trait, 100 reps of them. This is the perfect scenario, so let's make it as perfect possible. Let's simulate a heritability of 0.99. And then she ran it twice. A really cool thing about simple phenotypes is the user can specify a ballpark minor allele frequency of the QTN. So she utilized this capabilities. And so she ran this twice, once with low minor allele frequency QTN and once with a higher minor allele frequency QTN. And then, as I said, she considered 2 other simulation scenarios. So the really cool thing is that it is working. We are finding SNPs. So the left graph is when she ran this mixed random forest approach on all markers within 10 megabases of the QTNs. And then the right window picture is the one where she ran it within 20 megabases of each QTN. Now, the x-axis on both of these graphs is your base pair position on a chromosome, and then the y-axis is the particular chromosome in which the QTNs were simulated. Okay. So the red dots on each of these are the actual QTNs. The hashed dotted lines are, the respective megabase windows around the SNPs. And then the blue X's are instances where the QTN was, something within the vicinity of those QTN were identified. And we're not finding exactly the QTN because she removed that from the data set prior to running the analysis. Which is reality because we usually do not know what the QTNs are. But anyway, that concludes my talk. I'd like to acknowledge all of my Lipka Lab members and my colleagues both at University of Illinois and beyond, and anybody else I might have not included in these acknowledgments. So thank you, everybody.
But we'd like to thank you, Dr. Lipka, for joining us today. And we now have time for questions. Okay. So if anyone have questions from the audience, please.
Hello, Professor. Just a question about the random forest and the rare variants. Did you compare Talisa? Sorry, I
Yeah.
didn't know. You compare with the jewels. How did you, like,
Yeah.
is how much how did you set the additive effect or the effect of the kettle?
Yeah. Yeah. So, okay, this is a great question. So the question is, how did we compare this with existing approaches, right? So, Talisa already ran a simulation study where she used the unified mixed linear model, what I argue is the best suited GWAS model. And what she found from there is that the true positive detection rates out of 100 simulated traits was extremely low for a rare variance, but then it was much better for common variants. And so the immediate next steps that Talisa is taking now is she is running this mixed random forest approach on 100 replicate traits, and to quantify a similar metric.
Nice presentation. I have many questions, but one is, are the effect sizes that you explained in the first variance GWAS
Yeah.
correlated with the variance? Like, do you assume any correlation between the effect size, like the
Yeah.
mean of the SNP AA
Yeah.
and the variance?
Yeah. Yeah. So the question was, is there any correlation with the effect size and the variance? The cool thing is no. So, I mean, we simulated normal random variables where we specify the population mean and the population variance.
Independent.
The population variance was totally independent of the effect sizes of the G by E loci.
And another question, I want to know your opinion about those cuckoo plots. When you have
Yeah.
something which is clearly overinflated,
Yeah.
do you still consider the peaks as candidate genes, or do you discard the whole?
Yeah. So if we see a QQ plot that looks something like, and inflated like I showed for the real data analysis, should we look for candidate genes near those peaks? I would say no. Honestly, what I would do, I would do like a permutation approach or something like that to find empirical null distribution. And okay, I did not make it to that slide of the QQ plus enough time to answer that, but
Hi, Professor. It's a simple ask thing. I don't know if I understand right, but the key idea is cross genetic statistic data with not genetic statistic data. Or am I wrong? Because I am asking, because normally I know that we pick up the genomic data information and do a statistic works on. So you using genetic statistic data work on, and at the same time you pick up no genomic statistic data, how wave something about the soil, and another non-genomic, but continue being static data, but no genomic, and cross together. I'm right? I'm wrong?
so let me see. So is this question about the prediction accuracies that we observed in rice? Yeah. Okay. So yeah, I think. Yeah. So these results are definitely hot off the press. I think, yeah. So you were asking about environmental factors and soil factors and things like that. Yeah. So these rice data. So we actually use publicly available rice data on shoots of rice. So I think for these particular phenotypes, the soil and environmental factors did not matter, but I think, yeah, I think you have a point. I think that
Because the same molecular mark works in one way, the another way. So
Yeah.
you use this overall.
Oh, okay. Yeah. Yeah. Let's talk during the break. I think you're asking a very good question.
Okay.
Hi, Alex. One question related to the previous one.
Yeah.
on your QQ plot.
Yeah.
So one thing that I was wondering is, in a regular GWAS, those QQ plot, they are applicable when we are doing a test of 1 degree of freedom.
Yeah.
But here we are adding more than one parameter, because not only you have the covariate being added, which corresponds to 1 degree of freedom of a regular GWAS, but also the variance components that makes the residual heteroscedastic, because you're breaking the variances as well.
Yeah.
So could that be a reason why the QQ plot is overestimating the signal? Because the QQ plot is a 1 degree of freedom thing, but yours is like more of a 3 degrees of freedom type of thing.
I see. I never thought of this, to be honest. So like the differences in degrees of freedom, since you're looking at multiple environments, might be affecting. So the way that I'm thinking about this, is that a p-value corresponds to a null distribution, and like all p-values in the entire world, if the null hypothesis is true, follows a uniform 0-1 distribution. And yeah, this is a very interesting perspective that I've never thought about. So you think that a different metric than a QQ plot should be used to assess things?
Change the variance in the denominator for the p-value. So maybe instead of changing the metric, add more variability to that p-value because of the variation of the variances that he says.
That is a very interesting idea. Yeah.
Like, well, in animals we may have many random effects, not only the genetic,
Yeah.
the maternal. Each animal has its own environment, and we add that variance into the denominator, so that change a lot the QQ plots. Yeah.
Okay. Very, very interesting.
And then a second question on that very last slide.
Yeah.
I don't want to make you go all the way to the last one, but on the implementation of a random forest that accounts for the residual variance covariance of residual plus genetics.
Yeah.
That's a non-trivial implementation, I would add. Do you have any insight on how she got that rolling? Because you mentioned that your student implemented that from scratch.
Yeah.
Was that based on any previous implementation, or was?
Yeah. So there is a paper where this whole approach was developed, and it sounds like that it was developed in R, but then the R code was kind of orphaned. And
Thank you everyone that asked questions. Now, Dr. Lipka, the group would like to express our sincere gratitude for your lecture. As a token of appreciation, we would like to offer you a small gift.
oh, fantastic.